Before we do that, let's check in on our Socrative activity and see how that's going. Oh, first, by the way, the assignment that was due last night at midnight on Google Classroom, I said it was worth points. It wasn't worth points. My bad. My lie. But the reason that you were supposed to do that, and I'm glad that most of you did, is because that's another practice of me sending you a PDF, you annotating it, sending it back to me, because that's how the exams are going to work. For those of you that didn't do that yet, please do it, because I want to make sure that the process works for you before we get to the exam. If you did successfully complete that, I did leave you an individual private comment on the Google Classroom assignment. So you should go check to make sure that I said something. I gave everybody the same, I copy-pasted the same message. It said something like, I can see your edits, thank you, or something like that. Okay, so if you didn't get a message like that, that either means that it didn't work or you haven't yet turned in your assignment. Please do, so that I can check to make sure I can see how you've edited those PDFs. All right. Socrative, let's see. And again, getting a baseline, because I know a lot of you have learned some of these topics before, but not others, so it's the usual situation of it's a big class, there's mixed background, some of you really want to move on, get to advanced topics, but we're going to make sure we move together, which is why I want to know, when DNA is being synthesized, new nucleotides are added to the 3' hydroxyl. Perfect. The reason that this is interesting is because a lot of the annotations I got on the DNA replication bubble assignment don't necessarily reflect the understanding of this topic. They maybe reflect that you know this and you can answer a multiple choice question, but it's not coming through as clear when labeling is involved. And that's one of the reasons we're going to spend a bit of time in this class going over the answer to that exercise, all the labeling, where different enzymes are, where nucleotides are added, and so forth. But yes, 3' hydroxyl, answer C is correct. And I also had asked you to read one of the extension material papers for today. That's another thing we'll discuss by the end of class today or next class, depending on how quickly we move. ACS stands for, interesting. So yes, it does stand for American Chemical Society, but in the extension materials paper for today, ACS stands for. That manuscript did not say anything about the American Chemical Society. So yes, ACS does stand for American Chemical Society, but it's really Ars Consensus sequence. Okay, so that shows me that, I don't know, like at least a third of the class read the paper and maybe started working on those first questions, one of which was, what does ARS mean? So please read the paper before class. I know you've got a lot of things to do and you do have to prioritize. So it's understandable that some of you didn't, and that's why we talk about these things as well. So for those of you that didn't, we'll still get there. So here's the long list of vocabulary that we'll talk mostly about today. So before we get to that replication bubble assignment, I do want to give there's not just one last chance to talk about this, but since a number of you said that you had questions about the Meselson and Stahl experiment, this is the first experimental data we've seen in the class from a paper. I will definitely urge you, if you don't want to ask a question now, which is fine, you may if you want to, please come to my office hours. If you don't like the idea of office hours, maybe you think that that's where I sit in my office and I do my work. Think of it as consultation hours. Come talk to me about genetics, if you can. If you don't have time to come talk to me during office hours, email me and schedule a time. I can be flexible, but I have not had anybody from class come visit me, which either means I'm going glacially slow and you're all bored, or it means you need to come talk to me because you don't understand something in class. Over the course of this experiment, Meselson and Stahl had a tube there and a separate tube for 0.3 generations and a separate tube for 0.7 generations and a separate tube for 1 generation. So they were looking at snapshots in time of how the DNA content of the cells were changing. So that's all original. That's the all original DNA. And then we saw that as time progresses, we move from being all heavy to a situation where almost all of the DNA at the bottom is on the left side, which in this case, you can consider that the tube is oriented actually in this direction. So then you have the lightest, an overwhelming preponderance of all light DNA at the top of the tube. And that's just because the longer you let DNA replication happen, of course, the more DNA has been synthesized since you started the experiment. So over time, yes, eventually all of the DNA that the bacteria are generating is newly synthesized since the start of the experiment. So, no hands, move on. If you want more information, come visit me. Another question was, again, if nobody wants to own up to this, that's fine, we'll move right past it, but I wanted to give the opportunity to go back to the difference between conservative replication and semi-conservative replication, what the differences are and why we care about conservative replication. Even though we know that that's not how DNA replication works. Yeah.
Yeah, I kind of forgot the conservative replication model.
Yep.
If that makes sense.
So the concept of conservative replication is, we know semi-conservative, the two DNA strands, not all at the same time, replication bubbles, so this is a little bit misleading, but we know the two strands do separate and then serve as templates for the synthesis of, we should draw it like this, the second strand, based on base pair complementation. The conservative, so that's semi-conservative, and the only reason this is important is because of the Meselson and Stahl experiment, which was designed to distinguish whether semi-conservative or conservative replication occurs. Conservative replication would mean that that initial double helix is conserved. That is, it doesn't come apart and go back together again during replication. That is, those initial two black molecules stay in the same base pair double helix after replication, as opposed to being half of two molecules of DNA. So what would the other double helix after replication look like in conservative replication? It would be, yeah, this whole double helix would be new. Not one strand old, one strand new. You get one double helix old, one double helix new. And so, one of the things that Meselson and Stahl saw was after the first round of DNA replication, if you look in the figure, and we'll talk about this offline or individually later if you want to, what they saw was after the first generation of replication, all of the DNA was half original, or half heavy in their case, half light. In other words, these two molecules are sort of medium density. Each strand is one original, one new, half heavy, half light. So all of the molecules after one generation of DNA replication in bacteria look like that. One original strand, one new strand, and that's what Meselson and Stahl saw. If conservative replication had happened, they would have observed one entirely heavy, not half heavy, half light, one entirely heavy original double helix, a band that goes way to the bottom of the microfuge tube in sedimentation, and then one original, one all light, where both strands are newly synthesized. So that's the important difference between Meselson and Stahl's potential outcomes. This is the one they observed on top, semi-conservative. Could have been, and I don't really, well, since they didn't know how DNA replication worked, they might have had in mind that there's some enzyme, a big like donut-shaped enzyme, that the double helix just sticks itself into and it gets copied as the double helix moves through this protein. Right? They didn't know at the time, so they probably had lots of ideas about how these sorts of replication processes could happen in the cell. Semi-conservative happens, conservative doesn't. make this really clear. If that's the microfuge tube with some, I guess, red liquid in it, then the heavy, half-light strands would be here in the middle. They're intermediate density because they're half heavy nitrogen, half normal nitrogen. The all-heavy strands would go to the bottom. That's part of the conservative replication. And then if conservative replication had been happening, there would be an all-light band at the top. So those are the possibilities. Semi-conservative, after the first generation of growth, all of the DNA would be at a single position in this tube because it's all one density, an equal density. And that's what Meselson and Stahl saw after the first generation. Any other concerns? Yeah.
So I remember from the video it said like as the generations kept going on, it would get lighter and lighter. So in that tube, each generation is specific to each tube, so it's not going to like.
Right. We're growing this bacterial culture over time, and every once in a while they take out a sample of the bacteria and look at the DNA content in the bacteria. So over the course of that experiment, maybe we should talk about this later. Somebody asked about the end replication problem, and if you are here and you want to know more about it, we will talk about it, but maybe in context of this, which should be familiar by now. So I'm going to work through the solution to this, and then I've got a few examples, as you can see in the slides, of student responses. So first I want to sort of draw out the key to this, so everyone can hopefully be on the same page about how all of this labeling works. And then we've got a few examples that we'll collectively, anonymously review to see if we can find things that should be changed based on what we know about how DNA replication works. So does anybody have any questions about the labeling of the strands? Just move the nucleotides down from that double helix up top. And then I think it looked like everybody got the idea that there's, did I miss one? Oh, I missed, yeah, I put the first nucleotide at the wrong spot. That A should have been there. Oh well, it'll work. So everybody knows that, I think. By complementation, you draw the other nucleotides on the other strand. And shout out if you've got any problems, concerns, questions. How many TAs are there? 4. It's hard to keep everything straight, right? T, I think that's correct, unless I missed a letter. So that's what you should have done in one of the steps. So this is the replication bubble. This is, by definition, where's the origin of replication? There were some varying answers to this, but it's hard to say for certain where the replication origin is. It's somewhere inside the bubble. So any answer you put, as long as it's somewhere in here, is yes, this is the first spot where the DNA helix unwound, the double helix, so that defines this as an origin of replication. It's kind of a trick question. Most answers accept it. Somewhere in this replication bubble is the origin. This is small enough, it's only 12 nucleotides or so long, that you basically would call this whole bubble the origin. This is where replication started. Doesn't need to get more granular, more specific than that. Okay. There was a lot of difference on the polarity labeling. And so I had a lot of good comments from you with questions and concerns about this. So now's a good time to figure this out. What goes right there? Does it need to be, so I heard 5'. Does it need to be 5'?
Shouldn't matter as long as they alternate.
It shouldn't matter as long as they alternate. As long as you have 5' at one end of one strand and 3' at the other end of the same strand, it doesn't matter. But some of you did point out that there is this convention that we write DNA 5' to 3'. So most of you labeled the top strand 5' to 3', and then the bottom strand would be anti-parallel, 5' on the left and 3' on the right. That's fine, but the other way would be acceptable too. So you could put 3' there on the top strand as long as that's a 5', and then that would have to be, what here? If that's 5', that would have to be 3', and that would have to be 5'. So as long as you did one of those two things and were consistent, that's right. But now that I've drawn in the polarity, one thing that a lot of you missed, which is okay, not missed, didn't do, was labeling the polarity of the primers. So these primers are pieces of RNA or DNA?
RNA.
Primers are made out of RNA. And these are only there to start synthesis. We talked about last week that synthesis can only happen by adding nucleotides, the letters of DNA, onto the 3' end, and that's what we had in the quiz at the start of class. It has to be ended to the existing 3' hydroxyl group. So where is that on the top primer? The left side or the right side?
Right side.
So this primer,
The other side, because that's the 3' side of the RNA.
so this is the 3' end of the top strand. So that means the right side, the right end of the bottom strand, the RNA primer has to be,
That's the 5'.
this is 5'. 3' on the top strand is that end, so this has to be a 5' end, which makes the other end of that primer the 3' end. And vice versa on the bottom. That is the other way around. So hopefully this is okay. We've got, now, how many double helices do we have in this picture at this point? How many sections of double helix? Kind of 4. We've got one here where the replication bubble hasn't unwound the chromosome yet, one on the other end. And then we've got a small double helix, or duplex anyway, where each of the primers are. And that means that those primers have to be base paired according to ATGC pairing. So what's the sequence, let's say from 5' to 3' of the top primer? So we're going to start on the right side. First nucleotide in the primer is
A,
base paired with T, so it's got to be an A.
T.
This is where I screwed up, so I'm glad some of you caught this. This is an RNA primer. So instead of T's, there are U's. I totally would have forgotten that, except one or more of you actually remembered to do this, so thank you for reminding me before class. And then T, A paired to a U. So 5' to 3', that primer is AU, AU. AU? AU, pay attention. Sorry, bad joke. And then on the bottom strand, it's same. U paired with an A, T paired with A paired with U, T paired with A, paired with U. UA, UA, UA on the bottom strand. So now we come to the part where there was a lot of disagreement in how many of you filled this out from this point. I think the next question I asked was, where is the first nucleotide going to be incorporated as DNA synthesis starts? The nucleotides are added to the 3' end. So the first nucleotide on top that gets added by DNA polymerase, here's DNA polymerase, this big enzyme, gray shading. Big protein sitting on the DNA is grabbed onto the 3' end of the primer, and it's going to add what? What nucleotide is going to go there? It's going to connect to the 3' end, that 3' U.
Start with a T.
So it's going to be base paired to a T. So DNA polymerase, this big protein, is grabbed onto the 3' end of that primer, and it wants to add a new nucleotide there that will form a base pair with the template. So template is a T, so you have to add an A. And then RNA polymerase keeps moving to the left. that direction. And it keeps adding new nucleotides onto the end of the growing strand. So at this point, we've got A, U, A, U, A, and then you just keep base pairing. DNA polymerase keeps adding nucleotides by base pairing. So if we keep doing that, just to finish that strand, what's the next nucleotide? Base paired with A is a T, and then an A, and then a C, G, T, and so forth. Now, if we've done this correctly, let's double check. Always double check your work. This is not a math class, but still. If replication has occurred properly, then this double helix, this strand, should be the same as what? T, G, C, A, T, A.
Shouldn't be used.
Well, in this case, these are, now this is a good question, perfect question to ask. The primers are added, they're synthesized. The primers are made by an enzyme called primase. I didn't name it. It's easy to remember though. Primase only builds RNA primers. So it uses A's, C's, G's, but instead of T's it incorporates U's. But this DNA here, this is DNA, it's been added by DNA polymerase, the enzyme. So the only RNA bit in here is the primer sequence that's added by the different enzyme, primase. Once DNA polymerase takes over, all of those blue nucleotides are DNA, deoxyribonucleic acids. So T's instead of U's, because this is DNA now. Does that answer the question, or address it at least? Okay. So how long will that go on? This molecule of DNA polymerase, this enzyme, reading that strand and adding new nucleotides. Where does the process stop?
When it reads a stop code,
The DNA polymerase, this is just DNA replication. When you start talking about stop codons and so forth, that's later on in the central dogma. This polymerase will keep adding until one of two things happens. Where would it stop physically? This big protein sliding down the DNA, reading nucleotides and synthesizing new ones onto the second strand. What's this called? This bit here? This is the replication fork, where the DNA is all hydrogen bonded to the other strand. Each strand hydrogen bonded with its complementary nucleotide. What has to be happening there so that DNA polymerase can keep trucking down the nucleotides, right? So we can keep synthesizing DNA? This is an enzyme, absolutely. There's another enzyme called Helicase. So anytime, if you're not familiar yet, any protein or any word that ends in ASE, means it's an enzyme. That's just standard nomenclature for biochemistry. More than that, an ACE enzyme means it catalyzes some sort of activity. In this case, it's breaking the first part of the word.
The hydrogen bond.
It's unwinding the helix. The helicase breaks those hydrogen bonds and allows the two, and actually doesn't just allow to, it physically forces the nucleotides at the replication fork apart so that the replication fork grows. That is, the DNA keeps unwinding to the left. So first thing that has to happen at each replication fork, there's a molecule of helicase, and they unwind the DNA in both directions. Then DNA polymerase is able to come along behind helicase and read those hydrogen bond properties that are in the single-stranded DNA, and that's what DNA polymerase uses to match the right nucleotide by base pair complementation. So if helicase is doing its job, polymerase can come along down the top strand here towards the left and synthesize DNA all the way until it literally falls off the end of the chromosome. So if this was the telomere on the left side, if this is the last nucleotide on the chromosome, DNA polymerase just cruises on down and slides right off the end of the chromosome, and it's synthesized the very last base pair there. What do we call that? There's a definition for this that a lot of you are labeling your figures with. Strand nomenclature. Not polarity, not 5', 3', but there are two different types of strands in replication. Leading and lagging strand. So this is how leading and lagging strand are defined. If DNA polymerase can start at a primer, like here, and synthesize the entire rest of that molecule of DNA without stopping, that's the leading strand. And it's called the leading strand because it's a temporal thing. This strand replicates faster or first, depending on which way you look at it, because DNA polymerase doesn't have to stop. It doesn't need another primer. It's got the 3' end of a primer. It just keeps building DNA nucleotides onto each other until it reaches the end of the chromosome. So an important distinction between this and the lagging strand is how do we synthesize right now, if we consider this, basically from the origin of replication left, the top strand has been synthesized, replicated, base paired, complemented, whatever you want to call it. What part of the top strand hasn't yet been replicated?
Everything before that primer.
Everything on the 5' end of the primer. Everything down here hasn't been replicated yet because you don't add nucleotides to the 5' end. So there's no polymerase here going this direction, synthesizing the rest of the top strands complement. So what happens? How do we solve this problem? How does the cell solve the problem? What we need more, what do we need to start replication? We need DNA polymerase, but DNA polymerase can't work unless it's got a 3' end of a primer to start adding nucleotides onto. And that's where the part of this exercise asked about where is the next primer going to be synthesized. We need more primers to synthesize the rest of this chromosome. So you could imagine, in this case, that you add a primer say in red here on the top strand. So RNA primase, in other words, comes back, and it synthesizes another primer somewhere else in the replication bubble, like right there. So this primer, like the other primer that's base paired to the same top strand, has the same polarity, 5' on the right side, 3' on the left side. So now when you get another DNA polymerase enzyme comes and sits down there on the 3' end of the primer, just like before, which direction is that enzyme going to move? To the left or to the right on the top strand?
Same direction as last time.
Same direction as last time because it's adding nucleotides onto the 3' end of the primer, and it's moving from, in this case, from right to left. So the first nucleotide it adds in blue, the 3' end of the primer, it's going to add an A across from that T. Okay? Now what happens? There's one more nucleotide that has to be added.
T.
A T. Now what happens? DNA polymerase is stuck because it runs into a double helix. And there's no helicase there. There's no enzyme unwinding the newly synthesized chromosomes. That would kind of be pointless. So DNA polymerase gets stuck here. It's replicated or synthesized that last nucleotide before it runs into another section of double helix. So now presumably we've synthesized all of this from that first DNA polymerase molecule, all of this part of the top strand, second strand, and now we've synthesized from one end of the primer through where the origin of replication was. So this little chunk of RNA and a little bit of DNA on its end is the lagging strand. We also call that something else, named after the gentleman, I think it was a gentleman actually, prove me wrong, that discovered these tiny little pieces of DNA that were left after, as DNA synthesis was completing, the Okazaki fragments. These are fragments, they're little pieces of DNA that comprise, that is, they're composed of, they're made up of the primer, and a shorter bit of DNA that was added onto them by DNA polymerase. In other words, the leading strand is huge, usually, because it goes from the origin of replication until either the end of the chromosome or until you reach the next origin of replication, which we'll talk about in a minute. But these Okazaki fragments are short bits of RNA and a little bit of DNA that get synthesized. And the same thing happens on the bottom strand, just all the arrows point the other direction, because DNA is anti-parallel. So you could put a primer here in the bottom left, and then have DNA polymerase add those couple of nucleotides, and synthesis would stop there. So that should be somewhat similar, although in different words and in different circumstances than what was in the DNA replication video. But it's same, but different. You've heard it twice now, presumably. Now, we're not quite done yet. There are a couple other steps.
Why does the RNA start in the middle of the chromosome and have to deal with lagging strands and Okazaki fragments instead of just starting on the far right-hand side of the chromosome and doing one long leading strand?
Did it take too long?
I love it. Discussion. Finally. How many days of class? It's like the 12th class or something? Okay, well, it's probably because I talk too much anyway, but fantastic. So let me see if there's anything else that's, this is a good point to lead into the next topic, which is, it's a great segue. Now I'm ruining it by talking. What have we not done yet in terms of replication of the top strand? We've got this bit from the primer to the next primer, this leading strand from the primer all the way to the end of the chromosome. What's left to synthesize? What's not yet double-stranded on the top strand? There's one little nucleotide here. Once the helicase unzips those last two base pairs, there's going to be a couple of nucleotides there that are not replicated. Because where would we have to put a primer that would allow the replication of those? It would have to be out here in thin air where there is no chromosome. So RNA primase needs DNA to build the primer. This is the end replication problem. That is, the chromosome ends never get replicated all the way to the end of the chromosome. Because of this situation that we have where primase has to build a primer, but what primase needs is it needs some chromosomal DNA, the black strands. It needs something to build the primer onto, to base pair it with. So primase can't add a primer out here so that there would be a 3' end right there to add a G here across from this C to finish synthesis of the top strand. Every time your chromosome replicates, the ends get a little bit shorter because of this semi-conservative replication and the fact that there are leading strands and lagging strands. So then the question, and one of the answers, the question being, well, why doesn't primase So here's imagining that replication has just started and there are no replication bubbles. We've just totally unwound a chromosome, just for the purposes of illustration. Wouldn't it be great, and I guess I need to label orientation, 5', 3', 5', 3'. Wouldn't it be great if primase, now I just have to make sure I get my polarities right. Nope. Wouldn't it be great if primase, there we go, I'm getting there, wait for it, put a primer there, right at the very end of the chromosome on both strands. So what would happen? We've got the 5' end, of the primer at the very end of the telomere, the very last nucleotide of each strand. I'll work on my penmanship, I promise. So DNA polymerase could come along and bind those 3' ends of those primers and synthesize all the way across to the very end of each chromosome. That would be fantastic. Then we wouldn't have leading strands and lagging strands. We'd have one leading strand on each chromosome. Synthesis from one end all the way to the other. So that would be ideal, but there are two problems. Both of them having to do with the fact that this is not actually what happens in biology. It would be great if we could design the system, but we can't. Evolution designed the system. So what are the two issues? One is mechanical. Primase doesn't know where to build primers. It grabs onto single-stranded DNA and synthesizes RNA that's complementary to it. So RNA primase, in other words, just randomly picks single-stranded DNA spots on the chromosome to grab onto and synthesize a primer. So it might be that rarely it would actually happen to bind the very end of the chromosome and synthesize a primer there. That would be cool, but it doesn't. It's random. So sometimes it would build primers there, and other times it builds primers in between. That's one of the reasons we have the end replication problem. The reason we have an end replication problem is because, who said that? Thank you very much for whoever spoke up about the original response. It was, yeah, down here somewhere. I think it was a female voice, but I'm not sure. Back there? I don't know. No one's claiming. That's fine. The right answer, though, the second problem is time. And that brings us into the paper that we'll probably talk about next class, which is how long are our chromosomes?
A lot.
Millions or yeah, tens of millions of nucleotides. So let's just say that's 10 million base pairs. You could calculate how quickly polymerase adds nucleotides. How quickly can DNA polymerase stick nucleotides together to synthesize DNA? And it turns out, I don't have the answer on me, but we can do it as an exercise at some point if you want to, that if you had one replication bubble, say in the middle of the chromosome, it would take, I don't know, hours or days or weeks or something for DNA polymerase to start there and synthesize the two strands in both directions. And it would be, I don't know if it would be better or worse if the replication bubble was at the telomere. You'd basically have the same problem, is that DNA polymerase has to start at one end and continuously synthesize 10 million nucleotides, let's say. I can't imagine doing 10 million of anything, much less, although I'm not an enzyme, much less sticking 10 million nucleotides together and not making mistakes, right? We talked about balancing speed versus accuracy when we did the let's copy this DNA sequence by hand by typing what I projected on the screen. The same issue is that it would take too long, we would not exist, if we had one replication origin per chromosome. Instead, we have multiple origins of replication per chromosome. That's not true of all organisms, but mammals, higher vertebrates, most organisms that have linear chromosomes, it's true. So instead of what's drawn there, chromosomal replication looks more like this. I don't know what that looks like, but it's a chromosome that has multiple replication bubbles. Why are the replication bubbles different sizes?
Because it's just when they started and how much DNA they've already replicated. So the bigger the bubble, the longer that has been replicating on both sides.
Right, so you can think of bubble size as bubble age. The bigger the bubble is, the longer ago in the past, the DNA melted. The origin of replication broke the two strands apart. So this is a very, say, young origin that just now melted and became single-stranded, and replication's about to start there, but this big bubble is an old replication bubble that melted maybe hours ago And so there's been more opportunity, more time for DNA polymerase to synthesize these double-stranded bits of the replication bubble. So this is the other reason we have to have Okazaki fragments, is if we only had one replication bubble, and if DNA primase was useful and synthesized primers at the very ends of the chromosomes, we could have a single replication origin, except it would take a lot of time to do that. So the solution to that is we have multiple origins of replication on each of our chromosomes. The consequence of that is leading and lagging strand synthesis. Because you have to have a primer to start each little bit of synthesis. And so as the replication bubble opens, say over here, how are we going to synthesize that bit of the chromosome? As we saw before, that means that a new primer has to be added here to synthesize that bit of the chromosome, and so forth. So leading strands, there's always one leading strand in both directions from each origin, but they're always opposite to a lagging strand. Question?
So why is it the Okazaki fragments instead of just the old, or the other replication bubble catching up to the new one?
So both things happen, and that's a great question. So in this case, if we zoom in on that pair, for example, there's leading strand synthesis that will continue here until that DNA polymerase runs into this red primer. But we need Okazaki fragments to fill in the opposite strand. So this will be like that. But then that polymerase also runs into a primer. And this is how replication stops, in other words, is that two replication bubbles meet each other, and the polymerase enzyme runs into the end of an existing double strand, and it stops. So from each replication bubble, there are two leading strands going in opposite directions using the two different original double helices as templates, two original strands as templates. But then the other bits that are on the other strand from the leading strand have to be synthesized in yellow here as lagging strands. So in the end, then, every template DNA molecule is original. It stays the same. It doesn't break or anything like that. There are two parent strands of DNA. So at the very end of replication, we've got a bunch of primers in red, and we do need to label this. 5', 3', so that we know which are the 3' ends of the primers. 3', 3', 3'. Nope, I did that wrong. Absolutely right. So wait, no, the 3' ends are
The bottom ones were right
yeah.
at the top.
The bottom ones are 3' ends, anti-parallel, and this one, they're on the left sides. Thank you. So then those each get extended up until the next primer. So you get something that looks like a patchwork of newly synthesized DNA. Some of these are leading strands, some of these are Okazaki fragments. So the original two black molecules are still there. The parent strands, the template strands, the original double helix, doesn't do anything. It stays intact, the whole chromosome from one end to the other. But the newly synthesized strands are made up of RNA and red and DNA in blue. And you can see here what's happened is, at least I think you can see what happened. Did I do that correctly? No. What did I do wrong? I synthesized from the wrong ends of the bottom primers. You got to tell me these things when I'm testing you. At least I got to correct myself. What do I do here? 3' end is where you add new nucleotides, right?
Yes.
Okay. Thank you.
The 3' hydroxyl.
From the 3' hydroxyl. So what hasn't been replicated?
The very beginning.
The something. That bit of DNA on that chromosome, on that strand, and this bit on the other strand. Right? That's back to there were no primers there synthesized that would allow synthesis to occur. So our telomeres get shorter and shorter every time our chromosomes divide. Our DNA doesn't get replicated quite as much every cell division. Our chromosomes get shorter and shorter and shorter and shorter and shorter. And if you divide enough times, then your chromosome's gone. Fortunately, that doesn't happen to us, but it could if we didn't have other enzymes that help save this process. We'll talk about those maybe some other time. So 2 more ways that the cell needs to fix. There are a couple problems left before synthesis is really complete. Last 2 really important steps. One is, are our genomes DNA or RNA?
DNA.
Are your genomes RNA?
I sure hope not.
Our genomes are DNA. What's up here? Blue is newly synthesized DNA, red is RNA. We have to get rid of the RNA. Can't have any RNA left in our genome. So what happens?
Enzyme comes through and fixes it.
It's an enzyme. It's another protein that comes in and recognizes when RNA is base paired with DNA, and then it gets rid of the RNA. Literally, it like chews up the RNA and spits it out, and then adds in DNA to replace it. Seems like a lot of work, but that's how our cells are designed. So all of those bits of RNA get replaced with DNA, and then we have one more problem, and then we'll break for the day, and then we'll come back next time. And by the way, that means don't do anything else in terms of reading for the next chapter until after next class. So what it says in the slides due for next class, we're going to keep carrying on with this set. The last thing, there's one more problem.
What about the naked strands?
So
Ones that don't have any.
the replicated DNA just doesn't do anything. It really does. It folds up into a secondary structure that protects itself, but we'll talk about that later. Ask me again if we don't get to it. The problem is none of these pieces of DNA are connected to each other. This strand, this little bit of blue, the backbone isn't connected to that piece of synthesized DNA, isn't connected to that piece of DNA, isn't connected to that piece of DNA. These are all independent little short fragments of single-stranded DNA that are base paired to the template. So one more enzyme is necessary. Does anybody know? Oh, we didn't talk about gyrase. Ask me about gyrase next time. It's not gyrase, unfortunately. Ligase. DNA ligase comes in and fixes the phosphodiester backbone of DNA. It ligates, it ties together each of those little independent pieces of DNA so that it's all one strand of DNA from telomere to telomere. If it wasn't, if we didn't have 2 complete strands of DNA, then all hell would break loose. And maybe we'll see what happens when hell breaks loose next time.